Hi, welcome back. Today we're going to talk about Chapter 2 topics, and hopefully this will be a review of cell structure and function. In your prerequisite courses, you may have had some introduction to either bacterial cell types, but of course for human physiology, we'll look at eukaryotic cell types. And in this diagram shown here from your book, you can see that there are a lot of structures inside the cell. The most prominent on human cells is the nucleus. So a lot of the time we will be at the microscopic level, either looking at cell structure and function or tissue level function in order to understand human physiology. So today in Chapter 2, we'll do a review of the biomolecules. There are 4 of those. Just like we had 4 primary tissues from Chapter 1, in Chapter 2, the biomolecules, which is where we'll start, include sugars, fats, proteins, and nucleic acids. So we'll look at those and then we'll dive into cell structure and function. And in this diagram from your book, you can see the machinery inside the cell that allows the cell to do all of the things that the organism does, all of the requirements to say that something is alive, the cell is doing. So in eukaryotic human cell types, we have a division of labor. So these different structures inside the cell here on cross-section, they all have a distinct job to do for the cell, and those are listed here in green. So the main ones you're going to be responsible for are the nucleus, smooth and rough endoplasmic reticulum, Golgi complex, lysosomes, peroxisomes, mitochondria, and ribosomes. We'll talk about each of those individually as we go through this lecture. Okay, so to start off, let's just review some simple biomolecules. Organics mean that it's a carbon-based. We use organic like, hey, we went to Whole Foods and bought some organic food. That's not how we use this term in science. Organics are all carbon-based. So I know you probably haven't had organic chemistry, and that's okay. We'll just look at some simple examples and go through the 4 biomolecules, which are listed here in green. So you should know a little bit about each of these that I'm going to introduce here. Both of these carbon structures shown are in their gas form. So methane is a very simple 1 carbon structure with 4 hydrogens. It's completely saturated with hydrogens. Next to it is propane. It's a 3 carbon structure, and you might be familiar with that from cooking with gas out on your barbecue. You hook up the propane, and this is actually what this molecule looks like. Down below is a branched structure, also organic. Those green hexagons are all linked together, and those are sugars in their stored form. Typically, sugar running around your body and in your bloodstream isn't a good thing. Sugar is important for cellular energy, which we'll talk about in Chapter 3. But if the cells aren't using that sugar, excess sugars can be stored in the form of glycogen. So you can see organic molecules can be very large, branched structures and can become very complex. I've boxed a review in your textbook on page 22. Please make sure as part of your study, whenever you see these boxed comments, you turn to your book and read those specific items that are being called out to you in these notes. So these simple sugars, which we just saw branched as glycogen, first are monosaccharides. So they're single, simple sugars. And if you read this diagram from figure 1 in your book, from left to right, we start with glucose. That's a 6 carbon sugar. Fructose, right next door, is a 5 carbon sugar. So it takes on a pentagon or a house-like structure, whereas glucose has a hexagon structure. There's also galactose, deoxyribose, ribose. These are just a few examples of simple monosaccharides, and the body prefers these types of sugars for quick energy. So disaccharides can be 2 monosaccharides linked together. So here we've got sucrose, which is formed through a glycosidic bond between glucose and fructose. And again, we see our glycogen down here at the bottom, where we have a lot of those 6 carbon hexagon sugars all linked together. Glycogen is found in the liver and skeletal muscles. So if you are out and about and haven't eaten, then the body will break down glycogen and release these monosaccharides one by one from this branch structure in order to give you energy throughout the day. Okay, so fats or lipids. They're also carbon-based, but one of the things you might notice right off the bat, this is figure 2, these long chain fatty acids highlighted in yellow lack oxygen. So you see CH2, 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 no oxygen. That's one of the things that makes oils and fats not want to mix in water. So I think we're all aware that oil and water or fats and water, they don't mix. So these are hydrophobic molecules. And this is an example of a triglyceride. So we've got 3, that's where the tri part comes in, 3 fatty acid chains hooked together by a glycerol head group that's over here on the left in sort of a brownish color. One of the lipid molecules that we will use a lot is the phospholipid. This is a very unique molecule. It's going to be found in all cell membranes. This is where we've got the fatty acid chains. Again, if you look at the yellow, they have used a notation where the zigzag line is inferred that that's a carbon. So you don't see the CH2, CH2, CH2s, you see a zigzag line. So the yellow fatty acid chains indicated by number 3 on your slide, notice how there's only 2 of them. And then the number 2 position is your glycerol, just like in the triglyceride on the previous slide, but one of the chains is missing. We have 2 fatty acid chains instead of 3, and we have substituted over here on the left in blue, indicated by number 1, if you read this from left to right, is a phosphate group. Phosphates are readily soluble in water. So this molecule actually has dual solubility. It's got the blue phosphate group, which is hydrophilic, it's water-loving. And then you've got on the right these fatty acid chains that don't like water. So we have a hydrophobic and a hydrophilic region on one molecule. And that's very unusual, but cells are going to use this to their advantage. If we go ahead and we look at the space filling diagram of these molecules, we have 2 formations. When you put an amphipathic molecule, amphipathic meaning dual solubility, in water, they spontaneously orient themselves into either a bilayer. Over here on the left, you see 2 rows and water on either side, kind of like what a cell membrane barrier does. You've got water on either side. You have water inside the cell and water outside of the cell bathing the cells. And then over here on the right, you have this little droplet that you might have seen if you were waiting for your pasta water to boil and you added some olive oil. You'll see the little droplets of water, or excuse me, of oil. On the right, the micelle, which looks like a circle. Notice how all of those tails, those fatty acid chains that look like tails, they all point towards the middle. They don't like to be in water. They'll spontaneously move away from water. Just like oil and water, you see they don't mix. So these 2 formations, either a layer, a bilayer, or a micelle, which looks like a circle, will naturally happen when you add a phospholipid to water. So the sphere is touching the water. So the water-soluble portion with the phosphate group loves water. So the circles that are attached to the tails point towards the water, and then the fatty acid tails like to point away from water because they're hydrophobic, which means water-fearing. So these are the 2 orientations that naturally happen with phospholipids in water. And we can use those types of natural tendencies for these molecules to orient themselves in water in a little bit when we talk about the cell, and the human body is 65% water. So we'll come back to that idea when we finish talking about our biomolecules. Here's the third type of biomolecule. This is still considered in the fats and lipids. This is a cholesterol molecule. All cholesterol molecules have these 4-ring structures, and they are the base structure of all steroid hormones. We'll talk about steroids in chapter 6, and these tend to have a hydrophobic type of behavior as well. Okay, so for the category under proteins, all proteins are made from the 20 different amino acids. There are 4 examples shown here from figure 2.8 in your book. And amino acids are basically the building blocks of proteins. And most functional proteins in the body are going to be the doers. So molecules that participate in chemical reactions and things like that are typically protein-based. So the amino acids are assembled in the correct order in order to make a functional protein. So the amino acid is an amine group and a carboxyl group. Carboxyl is highlighted in red. The amine group is highlighted in purple. And that green R group is what changes, and that's what gives you the different identities of the 20 different amino acids. So number 1 is alanine, number 2 is tyrosine, number 3 is glutamate, and number 4 is lysine. Maybe you're familiar with some of these, but you can probably check out all 20 of them in your book. I've just got 4 examples here, and that green R group changes. So in alanine, you've got a simple methyl group, and then in tyrosine, if you look, it's got a big benzene ring, that big 6-carbon ring in there. So the R group is what changes, and the other 2 parts stay the same, and that's how we identify the 20 different amino acids that build proteins. So proteins are functional, and one of the things, they have to be folded into their correct position. So when you have your primary structure, it's kind of like stacking Legos. So if I gave you 20 different colored Legos and I said, build a tower, everyone's tower would look different. But then if I gave you instructions and said, okay, the red Lego first, then the white, then the blue, then the yellow, then everybody's tower would be stacked in the correct order. So in number 1, you see the primary structure, where you have amino acid 1, amino acid 2, amino acid 3. And they've kind of used a Lincoln log type of model here to show you the carbons and the nitrogens, and then you've got your green R groups that indicate the identity of the amino acid. So they're in a specific order. So we've stacked our Legos, made a tower. Now we need to make it functional. So in number 2, down here in the left-hand corner, the secondary structures, like that Shirley Temple curl, which is called an alpha helix, you might recognize that if you've looked at a molecule of DNA. The beta pleated sheet, kind of like a little zigzag if you've ever folded a paper fan back and forth. These are the secondary conformations that proteins will take on. And then over here on the right, number 3, this chain of amino acids has been folded a couple of times. So it looks almost like a keyhole. And there are some structures that hold this molecule in place. So in order for proteins to be functional, they need to be in the correct conformation so that they can do their jobs. Proteins can also act as enzymes. So in chapter 3, we'll come back to this idea and look at how certain proteins with enzymatic ability can run chemical reactions in the body and keep you alive. Our last category are the nucleic acids. Nucleic acids include DNA, RNA, and ATP. ATP is going to be the name of the game in chapter 3. So energy transfer is typically what ATP is involved in. And of course, DNA and RNA contain genetic information, but they all three fall under the same category. So they have a basic structure. They have a phosphate group, which we saw with our phospholipids, and they have that 5-carbon sugar that looks like a house or a pentagon, and then one of 4 bases. If you're talking about DNA and RNA and ATP, they usually have 4 different bases: adenine, guanine, cytosine, and thymine. A's and T's and G's and C's make up the genetic code for each of us, and we have an individual sequence, which is why we all are a little bit different from each other. So the basic structure is a phosphate group, a sugar, and then one of 4 bases, which are highlighted in green. And this is figure 12 in our chapter. Down here is ATP. It's a little different when you compare the base and the sugar. ATP, so adenosine triphosphate. With the big red circle, I've circled 3 phosphates. So ATP is designed for energy transfer. So the big red circle here in the lower right-hand corner indicates that triple phosphate bond, which is characteristic of ATP. And when those bonds are broken, energy is released into the system, and then the cells have energy to do work. So ATP is going to be our main currency. So when you eat food, your cells can't use that cheeseburger. They have to use the biomolecules and then convert them to ATP. So ATP is kind of a universal energy source across all organisms. So ATP will be our main focus in chapter 3. So we're just looking at structures right now and kind of getting our categories of biomolecules organized in our mind or in our notes. And then a little bit on DNA. I mentioned those 4 bases. They're shown here in green. RNA has an exception. RNA will substitute a T for a U, which is uracil. Uracil is only found in RNA. And in this diagram from figure 214, we're comparing the structure of DNA to the structure of RNA over here on the right. There's a few subtle differences, and just by inspection, you probably see that DNA is a double-stranded molecule. RNA is single-stranded. And you can also see RNA has uracils in the sequence, and DNA does not. So DNA is only found in the nucleus, and it's protected. It's basically the blueprint for you. And if we have any mistakes or errors in DNA, that's a mutation. Mutations cause cancer and other diseases, so we don't want DNA out floating around in the cell. It's protected by the nucleus, which we'll talk about when we get into the organelles. RNA, however, can leave the nucleus once it's been transcribed, and it goes out and it finds the ribosomes, another organelle we'll look at, and it initiates protein synthesis. So according to the blueprint DNA, RNA goes out and finds a machinery that's involved in protein synthesis. So structurally, it's a little bit different. Being single-stranded, it's skinny enough to find its way out of the nuclear pores, and the ribosomes can sit down on this molecule, and the RNA coding, because it's single-stranded, is accessible. So the U, C, A, G, if you follow the coding on the right here, will be translated into the amino acids that build proteins. We'll have a video on this so you can see all the molecules in motion. Okay, so let's go through our organelles. And your job here is to be able to identify these organelles and be able to tell me 1 or 2 things about their function. So I want to be able to identify the structure, how is it unique, and what does it do? So a lot of the times we're going to be at the microscopic level, looking at cells, which is going to be your structural functional unit for life, and their collective activities will dictate what tissues do, what organs do. So function a lot of time is going to be at the cellular level. So if we go inside the cell, we're going to be able to study the organelles. And from this microscopic view, you can probably see pretty easily the large round nucleus in the middle. It's a little bit darker than the rest of the cell. The cytoplasm that surrounds the nucleus, and then you can see a distinct border. These epithelials look a little bit cuboidal, a little bit squared, and we discussed those in chapter 1. So there's the cell membrane, the nucleus, and the cytoplasm that are easily seen on a light microscope like what we have in the labs. Okay, so from figure 15, you can get an overview of the machinery found inside the cell. And we will talk about these organelles individually. And the first thing I want to come back to is the cell membrane. So here we see those phospholipids forming a bilayer. So there's 2 layers. Notice the spherical phosphate heads are touching water. So they form a barrier. Inside of the cell, at the top, the cytosol has a jelly-like fluid interior. That's where all the organelles will be found as well. And then outside the cell is the extracellular fluid. So ECF and ICF will be the terms I use if you're looking at the board, or if you come to class and you see my abbreviations, I'll use ICF for intracellular, which is inside the cell, and then the fluid outside the cell will be extracellular fluid, which will stand for ECF. And this membrane barrier made of phospholipids is selectively permeable. So it's not a brick wall. We've got to be able to get nutrients and oxygen in, waste products and CO2 out, and things like that. So it's an exchange between the inside of the cell and the outside of the cell. So selective permeability, that definition is on page 8, I believe, in your book if you need to read that. And this membrane will be found on almost all of the organelles we're about to look at. So all of the organelles are going to have their own membrane barrier. But when you're talking about the surface of the cell, in this diagram, similar to figure 16 in chapter 2, we see the phospholipid bilayer, and they've separated it here on the left, almost like 2 pieces of bread being pulled apart. And you can see the phospholipids. They're highlighted in blue. And we've got some cholesterol molecules kind of interspersed. Cholesterol kind of gets a bad rap, but it's needed, it's necessary, and it's incorporated in the cell membrane to give it some rigidity. If you think of how a lava lamp looks when it first starts warming up, you get one big bleb and then maybe some little blebs pinch off from that. When they run into each other, they become big again. So that fluidity is kind of like what the cell membrane creates in the body. So the cholesterol, also shown in yellow, is just kind of interspersed in between the long tail fatty acid chains on your phospholipids to give rigidity. Now, these large purple molecules are proteins. They add function to the membrane. So now you're not just floating around in water. Proteins embedded in the membrane add function. And some of these proteins have sugars attached to them. So the red arrow is pointing to a chain of sugars being presented on the outside of the cell. So the top portion is the outside of the cell, and then the bottom portion would be inside the cell. So these sugars pointed out towards the external environment are things like if we were to determine what blood type you are. This is just one example, ABO blood typing. That is testing for the presence or absence of these sugars being presented on the outside of the cell. And they kind of hang on, or they're anchored to these proteins. This large protein in the middle, notice how part of the protein faces outside, and it's got another portion that faces inside. This protein helps bring things that are outside the cell into the cell and could possibly kick things out of the cell. So we'll talk about active transport and osmosis and things like that as we move through this first unit. So hopefully some of these ideas are review, but we're taking a much closer look at cell function. So we've got sugars, fats, proteins, all in one picture working together to form a functional barrier for the cell. So these types of functions with these large proteins, these large purple proteins embedded in the membrane, allow for transport. They can be enzymes, so some proteins have enzymatic ability. They can act as receptors, and we'll talk about signal transduction. So when a hormone arrives, how does the cell know how to respond? So we'll get into that. And they can also provide the cytoskeleton, and I will show you a picture of that in just a minute. So the proteins that traverse both faces, external and internal, ECF and ICF, are called integral because they're integrated into the membrane and they touch both the external surface and the internal surface. And then number 2 is a peripheral protein. So I've circled a peripheral protein. Notice how it's a little bit smaller, and that purple protein is only on the inside of the cell. It doesn't traverse both layers of the cell membrane. So that's something to kind of keep in mind when we start talking about function. And here's a fluorescent image of the cytoskeleton. So these are proteins called microfilaments and microtubules, and they act like a scaffolding inside the cell, just underneath that inner leaflet of the membrane. So within the ICF compartment, that fluid compartment inside the cell, it really has some substance to it. It's kind of jelly-like. And when we add a fluorescent stain to these proteins, and then under the microscope use a UV light, they glow like this. So you can almost see how it forms these hair-like railroad tracks. And the cytoskeleton not only gives support to the cell, it's a way of transporting substances here and there when you're inside the cell. So that's the cytoskeleton. And then here we go back to looking at some live cells. These are epithelials from a cheek scraping. And there's a filter that kind of creates this halo around the cell. This is called phase contrast. So these are live cells in saline solution. And just like our microscopes in the lab, we can easily see the nucleus in the middle. We can see that there's definitely some stuff inside these cells. We need a little bit more power to look at the organelles, but we can definitely see there's stuff inside the cytoplasm. And then we can see a distinct membrane. So each cell has its own border or barrier. So the fluid outside the cell is distinct from the fluid compartment inside the cell. So this is our first look at the nucleus. So let's dive into its structure and function. Again, it's the site of DNA and RNA. And the nucleus, since it acts as a vault for genetic material, it has a double layer membrane. I'll show you that here with one of the diagrams in just a second. So we'll look at the nuclear envelope, which is sort of like the membrane for the nucleus. It's a double layer. There are nuclear pores so that the RNA can leave and go find the ribosomes. And let's look at that on cross section. It's a little easier to see. This is figure 18. And you can see with the big red arrow here in this call-out box, nuclear envelope is a double membrane. So the red arrow is pointing at that double layer of a phospholipid that we looked at earlier. So this nucleus is impenetrable. You don't want any damage to your nucleus. That's where your DNA is, and that's where RNA synthesis occurs, and can cause a lot of damage if you have anything penetrating the nucleus. So there's a nucleolus, this dark core inside the nucleus, which is for RNA synthesis, and outside is where the chromatin's found. So inside the nucleus, you're going to have regions for RNA, and then the chromatin, which carries the genes and all the genetic material, is inside of there. And these yellow little bullets, they form pores. So in this call-out box, right under where it says figure 218, they've really enlarged the nuclear envelope. You can see the double membrane, but there's these large yellow pores. So these nuclear pores are proteins, and they take on kind of a barrel configuration to form kind of a whiskey barrel port. So things can leave, like RNA. When it's ready to initiate translation, the RNA needs to leave the nucleus and go find these little tiny purple ribosomes. It's another one of our organelles that we'll talk about here in just a second. So the ribosomes are hanging out by the nucleus, ready to start protein synthesis through the process of translation. All of our membrane-bound organelles are listed here. The ribosome is the only exception. Ribosomes lack a membrane, and there's a reason why. So we're going to go through these organelles that are highlighted for you. These are vocab terms, so if you want to do a flashcard with the name of the organelle on one side and then its function on the other, that's great. Homework 1 was geared towards having you practice drawing a few of these organelles. So homework 1 kind of stepped into some simple cell structures that should be reviewed for most of you, but if not, you'll need to study up a little bit more on human cell function if you haven't had this already in one of your other prerequisite classes. Okay, so from the nucleus, which is highlighted here in purple, we start to see this blue maze, this labyrinth of folded membranes. And this is the rough endoplasmic reticulum. It's rough because it's got all those bumpy little ribosomes bound to it. So under the microscope, researchers call this a rough endoplasmic reticulum. Reticulum means maze-like structure. Maybe you've heard of a reticulated python or something, and that has to do with the pattern, that maze-like pattern. So the rough endoplasmic reticulum has ribosomes. Smooth endoplasmic reticulum lacks ribosomes. So if we remember that ribosomes are participating in protein synthesis, then the rough endoplasmic reticulum with ribosomes can carry out protein synthesis. The smooth endoplasmic reticulum is smooth and it lacks ribosomes, which means it can't participate in protein synthesis. The smooth endoplasmic reticulum, even though it looks maybe a little bit similar, has a very different function. It lacks the ribosomes, but it does have the enzymes for synthesis of steroid hormones. And we looked at cholesterol earlier, and cholesterol is the base product for all steroid hormones. And the smooth endoplasmic reticulum is also really good at storing calcium. And we'll come back and look at that in more detail when we get to Unit 3 and talk about muscle function. Calcium is the trigger for muscle contraction. And if we move away from the rough and the smooth endoplasmic reticulum, we see this large green series of flattened, stacked, like pancakes, a series of membrane-bound organelles called the Golgi apparatus, and it's highlighted in green. The Golgi apparatus is going to participate as like the UPS of the cell. So the Golgi apparatus is going to receive newly made proteins. And we saw earlier that proteins have to be folded, and their 3D configuration is really important to how they function. So the Golgi apparatus receives these protein packages, it folds them, it prepares them for their final destination. You get sort of like a zip code added on there, so the protein is going to be delivered to the right spot. It's pretty amazing what all happens in this little distribution warehouse called the Golgi apparatus. Prior to the proteins being secreted out from the cell, if they're destined to work somewhere else, sometimes the cell needs the proteins that are newly made. So proteins can sometimes be destined to stay within the cell to do their work. But the Golgi apparatus, or Golgi complex, depending on which reference you're reading, it may say complex or apparatus, they mean the same thing. It's all tied into distribution of newly formed proteins. This diagram I had when I was a student, so I copied it from my book. I like it because it shows from left to right the nucleus and how the rough endoplasmic reticulum is an extension of that nuclear membrane. So you're immediately taking the RNA that's been transcribed from DNA, and the RNA is going to sit down and find a ribosome, and you can immediately start translating that genetic code into protein. So the connection between the nuclear membrane and the rough endoplasmic reticulum, that's not random, that's intentional. So it's a great example of structure and function. So the ribosomes will start reading the RNA codons and translating the genetic message into amino acids. So we'll start stacking those Legos and making a tower, so making the primary sequence of the proteins. And then down here at the bottom of this diagram, you see the arrows and the big red circle. So proteins are being transported in these little transport vesicles, these little membrane-bound transport vesicles, kind of like a bleb on your lava lamp. And it's running into the backside of the cis space of the Golgi apparatus. And the Golgi apparatus is going to receive those newly made proteins. It's going to package and process and put a zip code on these proteins and just get them into their final folded configuration. And then on the right side, the trans space will bleb off, just like a lava lamp. You kind of have a little bleb coming off, another little secretory vesicle that's taking the ready-made proteins out to the surface of the cell. So from the plasma membrane, these proteins are being expelled. So their job may be to go outside of the cell and do their work somewhere else. Not all proteins are going to be secreted because, like I said before, some proteins need to stay with the cell to do their work. We saw proteins embedded in the cell membrane to add function. So that would be an example of that. Okay, so to create ATP from the foods we eat, we need to look at the mitochondria. Some tissues are going to have a lot more mitochondria than others, depending on the energy demands of that tissue. So this is a look at a mitochondria, and it's been cut in half, so it's on cross-section, and we can see inside. So it's a membrane-bound organelle. It's a double membrane, and there's this matrix inside, like a maze. So it's really packed full of even more membranes that are going to have proteins bound to it. So in chapter 3, we'll really look at function of the mitochondria. But for now, the foods we eat are going to be processed through glycolysis if you eat sugars, or lipolysis if you're on fat diet. And proteins can also be used for energy, but the conversion of foods into ATP, we're going to break it down into a 3-step process. So we'll have glycolysis, the Krebs cycle, and that occurs inside the mitochondria. And then the third step, the electron transport chain, which is located on this matrix. So we'll have a little bit of chemistry and look at energetics on how we actually make an ATP. Remember, that was a triple phosphate. So that triple phosphate portion of the molecule is where the body can extract energy. It's a great way to store energy in those triple phosphate bonds. Okay, so now we come to our unique organelle that lacks a membrane. So this is a look at the ribosome. This is a computer-based model. They took the protein sequence and formed a 3D model, and it kind of looks like a donut. You've got a large subunit on the right and a small subunit on the left, and it forms kind of a donut configuration. That hole in the middle is where the RNA will feed into the ribosome, and the ribosome is just going to thread down the length of the RNA molecule, reading the codons and converting that genetic information into a protein one amino acid at a time, stacking those Legos. So this video link should put a lot of these organelles into motion so you can see function. Sometimes the videos are a little more helpful than reading the book. Some of you, if you're visual learners, the videos will save you a lot of time and be very helpful. So this is all participating in translation from RNA to protein. Here are the lysosomes and peroxisomes. These may be a little more unique. You might not have come across these before, but let's just talk about them real quick. These are small little organelles. You can see that they're membrane bound, and they've definitely got some stuff inside. So their function is to clean and detoxify the cells. Sometimes the cell will die if these organelles degranulate to eliminate foreign substances. Sometimes the cell will die itself in the process, and that's okay. It's a trade-off to get rid of a foreign substance and lose a few cells that are protecting the rest of the body. So lysosomes contain hydrolase, so you can use the water to hydrolyze and split certain foreign invaders. Peroxisomes down here on the left has a purple crystalline core, and you really wouldn't be able to tell the difference between a lysosome and a peroxisome under the microscope. You would have to kind of see what's inside, what substance they carry. So the enzyme oxidase and catalase, if you had microbiology, you probably tested your unknown bacteria for oxidase and catalase positive. That would help you determine what species you have. So that's a hydrogen peroxide enzyme that can also destroy foreign invaders depending on what type of bacteria it is. So these are just some innate immune substances, I guess you could say. So each cell has a little bit of a defense mechanism. Okay, so this portion of the notes is going to look just like your biochem review in the Getting Started folder. So most of you should have an idea of what you're getting into in this class. Protein synthesis should have been covered in your general biology, which was a prerequisite for this course. I've included some videos, and if you need to review protein synthesis, so DNA to RNA to functional proteins, this is where you can do that in these notes. And hopefully this will be a good starting point for us to discuss mitochondrial function when we get to chapter 3.